SNP testing is rapidly becoming one of the most important tools for the genetic genealogist, as advances in the technology of SNP testing increase the power of YSNPs to create genetic descent trees. This presentation is a re-recording of a talk given at the recent Who Do You Think You Are live event in Birmingham in April 2016, with a few new or revised slides. The talk has been divided into 3 parts for this presentation. First, this part will look at the basics of SNP testing. Part 2 will examine some more advanced issues surrounding SNP tests and how to make best use of their results. And the final part will show how the results of SNP tests can be combined with more familiar STR tests to generate more powerful and detailed descent trees for a family line. The Y chromosome is used for understanding descent in the direct male line, as it is received by sons from their fathers, and lets us look at descent in this line all the way back up through human history. Most genealogists, though, are interested in their historical ancestry, and the Y chromosome is a great tool for researching the histories of surnames, since these are generally passed down along with the Y chromosome in the same way. Here we can see a map showing the distribution of an interesting Scottish surname, Cumming, in the middle of the 19th century. And here is the same surname around the same time in Ireland, showing an interesting distribution that might suggest a connection with settlement around the time of the plantations. While this map, for the very similar surname, Cummins, shows a very different distribution, suggesting that there may be more than one origin for these names in Ireland, and DNA can help explore these origins. We will be coming back to the Cummings later in the talk. So genealogists who have tested their DNA will be familiar with a type of mutation known as STRs, short tandem repeats. Short sequences of DNA repeat themselves certain numbers of times, and these repeats can increase or decrease, producing subtle differences on the Y chromosome. These differences can be tracked and compared to spot possible relationships. We can see the results table here showing numbers of repeats at the tested positions, and the coloured cells show the mismatches. The relatively small number of mismatches shows that these people are likely to be related. And here are some of our Cummings friends. Although we can see their likely relationship to each other, it's hard to construct a tree from this data that demonstrates how they are related, so SNPs, single nucleotide polymorphisms, come into play. These are single mutations at one point in the DNA sequence. Here we see an example sequence, and we'll take a little look at this C here in the middle, and let's suppose it changes to a G, so that we now get a G in its place in the sequence, instead of the C that most people have. This creates a change or a mutation in the DNA known as a SNP, from C to G. This is a well-known SNP that many of you will have if you're in the R1b group, and is widely known as L21. So until comparatively recently, STRs were seen as the main tool for family and surname history research. That's in the historical period, while SNPs were seen as being mainly about deep ancestry, our prehistoric origins or ethnicity. Many people try to find their terminal SNP, the most recent group-defining SNP they had, but most of these would still generally be found as having occurred in the ancient or prehistoric times. New tests, especially the ones known as next generation sequencing tests, or NGS, are making SNP testing far more powerful for looking at more recent descent and relationship too. So NGS testing is one of 3 types of SNP test that you can take. Sequencing tests read long sequences of your Y DNA and read all the mutations on those sequences, SNPs and STRs. They read the unmutated positions too, so you can compare your full sequence with another person's. And most importantly, they discover new SNPs in your Y that have not been found before. It's possible to test single SNPs as well in a one-off test known as Sanger testing. This is how most SNPs were tested before sequencing tests became available, but it can be cumbersome and expensive to test many SNPs this way. The Sanger tests, however, can be the most reliable SNP tests you can take, so they are excellent tests for doing confirmations of new discoveries made in a sequencing test. And now it's possible to test several SNPs all at once, in panels or packs of SNPs. Maybe there's a set of SNPs that relate to your surname or the wider group you're in, and these let you test them all in one go. While quicker than single SNP testing and cheaper than NGS, it's important to remember these tests cannot discover anything new or unknown. They only confirm whether already known SNPs are present or absent in you. So a quick look then at the main companies who offer YSNP testing. Family Tree DNA offer all three forms of test, with a sequencing test known as Big Y, along with a wide selection of SNP packs and single SNPs. Full Genomes Corp offer their own sequencing test, YElite 2.1, and YSeq are specialists in single SNP testing, also offering a wide range of SNP panels, single STRs, and they give very useful advice to testers about newly discovered SNPs, whether they are Sanger testable or look good stable candidates to be new SNPs, and so on. If you take a sequencing test like Big Y, you may find it a good idea to take your results file, particularly your raw data, to groups who offer third-party analysis of the data. It's complicated, and converting the results you receive into a readable descent tree takes some work interpreting the raw data. So first we'll look at some of the commercial analysts. YFull.com have a user-friendly graphical interface and help testers to read easily the most important aspects of their results for a small one-off fee. Their online tree is available free for everyone to consult, though only contains results that have been submitted to them. As well as doing their own test YElite, FGC also analyse results from other companies' tests for a similar cost to YFull. Which one to choose may depend on your haplogroup, YFull being particularly good for R1A, Q or I haplogroups, while FGC have worked closely with some of the R1b subgroups, for example. Clarify Y-DNA is another analyst developing an online tree and discovering new SNPs in raw data. And for anyone who belongs to the very large R1b P312 subgroup, submitting your results to the big tree is an absolute must. This is not a commercial group, but is all done by one volunteer, Alex Williamson, and is free, and has created the most detailed online tree yet based on SNP results. Here we can see the top line of the tree showing the groups just below P312, including the L21 we looked at earlier, and you can go to see the full Y tree on the URL given there. If you take a SNP test, you really should join your own haplogroup project at Family Tree DNA. The volunteer administrators there will often be the true experts on their and your particular part of the Y tree, so their advice is invaluable to you. Now, I've referred to L21 as the name of a very common Y SNP, so this brings up the question of how we label and refer to SNPs. In truth, there is no standard way to name new SNPs. This is somewhat of a chaotic area. Any new SNP will at first be identified as a long number, which stands for its position on the Y chromosome. There are about 59 million positions on the Y, so these numbers function like coordinates. This is followed by the original value at the mutated site, here a T, plus the mutated value, here a C, which are referred to as the ancestral and derived, i.e. mutated, values. There are slightly different ways of writing this down. We see two examples here. In practice, names are given to SNPs which are shorter, easy to remember and say, and are usually combinations of letters and numbers. But they can often be as unmemorable as the long numbers they're meant to replace. You may see in your results SNPs named with prefixes like these, which were found in research labs in academic studies. And names are also given by the analysts, the analysis companies, or citizen scientists, ordinary people like you doing their own research. Here are some examples of some of the common prefixes found today and who is giving names using these prefixes. There are others. Be very careful when you're looking at named SNPs. It's becoming clear since the big Y got going 2 years ago that many named SNPs found in older studies are not looking reliable or consistent markers of branches in a tree. Some of them are, many of them are not. So names for SNPs do not always confer significance. This is another area where advice is essential to you to interpret your results. And I would argue that oftentimes SNP names are given too quickly. If a SNP is private to one person and not discovered in anybody else, then the coordinate plus mutation pattern works fine, and there's no real need to give a name to this new SNP until someone else is found to share it, or you want to do Sanger testing to confirm its status. If you're running a SNP-based project, you should compile a catalogue of SNPs discovered in your tree. Here is an example from one that I've been building, giving all the names found and all the duplicate names, there's one case we can see here, plus all the coordinates and the mutation values of the SNPs we're discovering. This means we can look up any of these using any of these information, position numbers or names or duplicate names. I'm also keeping a note of the quality of the SNPs found. So the stars given here are from YFull.com, that's their system, and we can see there's one here that has just 1 star. This is of much lower quality than the others, and in the long run it probably will not be acceptable as a good reliable SNP. The YBrowse site is a superb online SNP catalogue being maintained by Thomas Crane of YSeek, and you can use this to look up any new SNP you find in your test. Many labs are now doing this themselves before awarding new SNP names to make sure they're not already named. However, multiple naming of SNPs is still going on and can be very confusing, so again, watch out for that. Here's a view of a YBrowse search window. You can see the position of the SNP here in the top left-hand corner in the search box, and here in the upper middle you see a diagram of the Y chromosome showing the position which we're looking at very approximately, and then lower down you can see that we're looking at the SNP we mentioned earlier, L21, and this has 2 more names, although L21 is the most common name it's known by, and also down the bottom we can see that this SNP is also listed in the academic database DBSNP, identified here by an RS number. You can also on this page zoom out to see more of the DNA sequence around your SNP and see what other SNPs and STRs there may be in the neighbourhood of your SNP of interest. Another excellent and free resource is the online tree developed by YFull.com, and this has an excellent online search function which allows us to examine named SNPs in more detail. So we'll do a little test. Here's one I prepared earlier. a SNP called YP984 was very important to one branch of the Cummings surname. So we can click on search, and this will bring up the YFull search here, and we can see that this SNP so far occurs only once in the known human Y tree, and that's good, as it can be therefore a reliable branch marker not found in multiple positions. So if you have this SNP, you can be pretty well sure that you belong in this branch. And clicking on the search result here will take us to the position of YP984 in the YFull tree, so we can see more detail about where it is and which other SNPs are ranked along with it in the tree. So let's look at another one. This is another SNP, Z3382, which I've chosen at random, and we search on this, we get a very different looking result. This is much less consistent because this SNP is now occurring in many different positions on the tree, all of them in different haplogroups. And in fact, what we see here is just the beginning as this continues off down the page. This looks very suspicious. It suggests this SNP may be inconsistent or unstable and not a reliable branch marker. Indeed, YFull rate this as only a 1-star SNP, and to them it's not worth placing on the tree at any of these known positions. So again, we need to watch out that even named SNPs can be dubious. We need to research a little bit to find out whether any named SNPs we have are actually good quality branch markers. So moving on then, we'll look at what happens when you take a big Y test, one of the two NGS tests, the one run by Family Tree DNA, or if you arrange one for a male tester in your family group, what do you get for test results? You get a number of screens of results. We'll see a couple of examples. This one shows the list of the named SNPs that are positive in your test. Again, watch the warning above because many of these may not be good or reliable SNP calls. We can see here that several of these are marked as no in the column on Y tree. That is not a good sign, doesn't offer confidence that they're of good quality. On the other hand, these ones, all on the Y tree, these ones are all good quality. These SNPs here have all been discovered in Big Y since 2014, and these are known to be good, reliable branch markers. You'll also have a screen for your novel variants, that is, your private mutations which only you have, and are not found in any other person so far. At least that's the theory. Again, we can see on this screen that this person has 138 of these novel variants. Except many of them actually are not novel at all. They're already known, and they're already on the Y tree and other people's results. In actual fact, most people can probably expect to have between 2 and 20 genuinely novel variants depending on where they are on the Y tree and who close to them has been tested already. But if you do all of this, what actually does it offer you as a genealogist, a family historian, or a surname researcher? Well, the objective of all this is to develop a more detailed descent tree than you may have had previously through genetic testing. This one here shows a subgroup of R1A, which was unknown before Big Y testing took off 2 years ago. So all of these SNPs have been discovered and arranged on this draft experimental tree since then, since spring of 2014. At the top, YP355 probably appeared in Norway, maybe around 600 BCE or so, and gave rise to these several branches below, which are now found in Scandinavia, Finland, Germany, England, Scotland, and Ireland, and Wales. And from approximately this point downwards, we're now looking at SNPs that have emerged in the historical era, since early medieval times. Remember, SNP testing used to be thought of as being about deep prehistoric or ancient ancestry. And if we take the line down to round about here, then now we're looking at the time that surnames became inherited, around 1200 to 1300 or so in England. The SNPs below this line belong therefore to the so-called surname era. and some are even very clear markers of particular families or surnames. If we look at the corner of the tree down here, we can see that we're back with our friends the Cummings, and with their associated surnames Kemp and Jacobs. And if we look at this tree, we can see that many of the SNPs come in blocks together. Here, for example. And these blocks of SNPs always seem to occur together with each other. Each of them, in fact, must have appeared in different generations. But the many descendant lines have then died out, or maybe they've daughtered out, or maybe they just haven't tested yet. So maybe one day future tests might split these blocks up into a more complex tree, but SNP blocks have become part of the scene since Big Y. So you may find in your test you'll get many SNPs marking branches of your tree. You'll just as likely find you've got many blocks of SNPs marking branches of your tree. And some of these SNPs, however, are defining their surname families. So YP992 and all the SNPs below it only appear in this Cummings family. YP1414 and the ones below it also only appear in this family of Kemp from Cavan in Ireland, although they've only done 2 tests so far, so taking more tests would probably be advisable to confirm this particular finding. So this is what SNP testing is offering today to researchers, the hope of discovering SNPs in the historical era which can define surnames, which can define your surname. So to sum up the first part of the talk then, here is a strategy for doing SNP testing for a group of Y chromosome testers that's already been defined by STR testing. First, you want to conduct some NGS tests, as many as you can afford, preferably at least 2 different looking testers, more if you can afford it. Then take your results to a third-party analyst or to your haplogroup project to help sort the results into a logical descent tree. Third stage, you should consider using Sanger testing to retest any of the more marginal or questionable SNP calls, or any private SNPs you may have that you want to explore further. Since Sanger testing is a different methodology to NGS testing, it's a good idea to check marginal calls with the second technology to make sure that these marginal calls are in fact reliable. And then finally, when you have a good set of confirmed new SNPs for your group or surname, you can approach one of the companies to see if they would make a custom SNP panel for you. This will allow other testers in your group to check their DNA for your group's SNPs much more cheaply than taking an NGS test, especially if you no longer need to prospect for new SNP discoveries in your group. In part 2 of the talk, we'll now look at some of the more complex issues around NGS testing, and then we're going to look at how detailed trees can be built from new SNPs and old STR data.